Hey everybody, we're approaching the end of the semester, and before we finish up, I just want to talk to you a little bit about evolution. I know I've talked about it a lot as we've gone on, but there are some things that I have not mentioned yet, and I want to hit those because I expect you to see a lot of evolution on your exam. Evolution is the change in the genes of a population over a long period of time. So there's natural variation in a population, and some individuals are going to have different traits or stronger versions of traits that will be more beneficial in their environment, and they will live longer, pass on those traits to more offspring. So you should see those traits accumulate over time. That's the basic idea of evolution. Now, these are the five mechanisms or forces. Natural selection is the one we're going to focus on, and that's what I was just kind of describing to you. We've talked about genetic mutation and recombination through crossing over, and I'll also mention gene flow and genetic drift. Gene flow occurs when members of a population migrate around, interbreed, and pass their traits on to future offspring. So every allele that an organism of a species possesses goes into the gene pool. Um, and all of the alleles are available to the population, even if they're not expressed. So in a heterozygote, you have the dominant and the recessive allele. The recessive allele is not expressed, but it is there to be passed on to offspring. So it is part of the gene pool. It is available. So mutations are changes to DNA or RNA that are going to code for an amino acid sequence that makes up a protein. And if you get a change to that nucleotide sequence, it can possibly change that amino acid sequence and therefore the structure of that protein. So changes to DNA are more permanent because you keep DNA for the life of the cell. Changes to RNA are less permanent. So if there's just a mutation in the RNA, it can be degraded and you can make more RNA to make that protein again in the correct way. So mutagens are things in the environment that cause change. Gen for beginnings, muta for change. These are some of the mutagens that you might come across that could cause these changes to the nucleotide sequences in your nucleic acids. There are two kinds of mutations. We have gene mutations and chromosomal mutations. Gene mutations affect one single protein. Remember, gene is a small piece of DNA that codes for one protein. But chromosomal mutations can affect several genes at once or several proteins. You can review these if you want with your mutations lecture, but there are three kinds of gene mutations: substitution, where you switch a nucleotide; insertion, where we add a nucleotide; and a deletion, where a nucleotide is removed. Um, Now, a substitution can only change one amino acid, a good way to get genetic variation within a protein sequence. So these are less harmful than something like an insertion or a deletion. Usually, insertions or deletions will throw off the reading frame of the ribosome, so you won't get the same three-letter code for these amino acids. Remember, there are different types of substitutions: your nonsense, missense, and silent. Silent, no change to the amino acid sequence. Missense is a change to a single amino acid, like you see here. And then a nonsense is a code for an early stop, which makes a protein very short and it can't do its job. We have some chromosomal mutations. So this is a chromosome that's been divided into regions, and there are many genes in each region. When you take one out, it's a deletion; double one, a duplication; flip one around, it's an inversion. So BCD is now CBD. Or translocation, you're taking a piece from a non-homologous chromosome, something completely different that doesn't match with this chromosome at all, and inserting it into a different chromosome. So these genes will be misregulated, and this is how we get certain types of cancer. These are examples of non-disjunctions. Remember, that's when you get too many or too few chromosomes in a gamete, an egg or a sperm cell, and that occurs during meiosis. Big one that you need to know is having 3 of chromosome 21 is Down syndrome. There are several abnormalities to sex chromosomes that one could possess and be fine. Um, Trisomies, you can have 3 sex chromosomes or you can just have a single X chromosome. This is the only monosomy that a child can be carried to term with. Any other monosomy where you're missing one of these chromosomes would lead to a miscarriage of the embryo. We mentioned genetic recombination when we talked about meiosis, or the making of gametes, egg and sperm. So this is crossing over. You have your homologous chromosomes that match in terms of what genes they carry, but may be different in what forms of those genes. And mom and dad's chromosomes just switch a little piece. So the genes that are on these chromosomes are no longer linked together in the same way. And that's one form of genetic recombination. You can separate these groups of mom and dad's chromosomes in different ways into different gametes, and that's called independent assortment. So you probably are not going to separate all of the genes you got from your mother into one gamete and all the genes you got from your father into a different gamete. Some of your gametes will have more chromosomes from your mother, some will have more from your father, and that's what we call independent assortment. And then the combination of different egg and sperm between individuals carrying different genes will introduce different combinations in offspring as well. Genetic drift, I've only barely mentioned. We talked about this with the tigers. When a small population loses genes due to random chance, and we actually are seeing little genetic drift in the tiger population because of those corridors that allow gene flow between members of different populations. But genetic drift can occur when you have very few individuals and some of those individuals just die. It's not because they have traits that necessarily are bad for them, it's just they happen to not make it. So those genes that they possess are taken out of the population and it becomes less varied. So that's called the bottleneck effect. And we see that when you have few genes remaining in the gene pool, lack of diversity. This is Charles Darwin. Maybe you recognize the name. We call him the father of evolution. Though the idea of evolution, that populations change over time, had been around a long time, it wasn't known how these changes accumulated in populations. So Charles Darwin came up with the theory of natural selection. This is the main mechanism of evolution. Though genetic recombination, gene flow, genetic drift, and mutation do go into evolution, natural selection is the main way we see the accumulation of these traits. So Darwin traveled widely, collected a lot of fossil data, and the data that he collected he used to write this book on the origin of species by means of natural selection. He wasn't going to publish it at first due to some of the beliefs of his wife. He didn't want to offend his wife, and so he kept it a secret for a long time. But he eventually received a letter from another man named Alfred Wallace, who doesn't get as much credit. But Alfred Wallace was studying organisms in a different part of the world, and both of them came up with the same theory. When Darwin saw Wallace's letter to him, he knew he better publish quickly if he wanted credit. And so that's why Darwin became known as the father of evolution, and his book was much more detailed than Wallace's paper. He had worked on it a lot longer after all. So natural selection is the process where organisms best suited to their environment survive and reproduce. There are 4 principles, and they're all illustrated here in this picture. We have an overproduction of offspring. There are more of these beetles born than can possibly survive. So a lot of them get eaten very quickly. We see this a lot with our selected species, like fish and frogs and insects. There is variation in this population. The variation here is in the color. So we have green beetles and we have brown beetles. One of these variations is an adaptation. It is a trait that helps this organism survive better in its environment. Therefore, it will survive longer and it will make more offspring. So we see that the brown color is the adaptation because these birds prefer eating the green. So when they see the green beetles, they're taken out of the population. They will not pass on these traits, but the brown bugs will survive longer and reproduce more. And that's going to lead to descent with modification. Throughout the generations, we'll see this brown trait appear more often in this population. So natural selection operates on genetic variation, these differences that we see in a population, and the individuals with the most useful traits are going to survive better and pass on their traits to the next generation. And those individuals who make more offspring with those beneficial variations are going to be more fit for their environment. Some traits may be inherited from mutations, those changes to DNA, we've seen that before. And that's how these variations are going to arise. And some of those traits will be good traits and some of those traits will not. And those traits that are not beneficial will be removed fairly quickly from the population most of the time. Of course, if they are recessive, they can then hide and be passed on to future offspring, like we see with cystic fibrosis and Tay-Sachs and some of our lethal recessive alleles. I mentioned that organisms with traits that are more beneficial for their environment will survive longer and make more offspring, and those organisms are more fit. You often hear survival of the fittest, and we think, oh, that's the strongest that's going to survive, but not always. What we mean by fitness is an organism's ability to reproduce. So you could have a big, strong male lion who is sterile, and he would be less fit than a weak, scrawny lion that can actually bear offspring. So helpful traits increase fitness if they help this organism to survive longer and reproduce. And only the most fit organisms will survive in their environment, and successful adaptations will be passed on with increasing frequency. You'll see more of them as time goes on. There are two main outcomes of natural selection. Either a population is going to continue on and develop and change, maybe even stay the same, and you could get a split between a population and speciation, or a population is going to go extinct if it can't survive in its environment. So let's look at these salamanders a little closer. Geographic isolation is one of the main ways that speciation is going to occur. There's some physical barrier that's going to separate two populations so that they're no longer able to pass on any traits between them, and any mutations that arise in one population will be retained there and not passed to the other population. So those mutations and genetic recombination can accumulate and you get a different species. So we'll look at this example of the salamanders. This is our ancestral population living here in Washington. And as this population's growing in number, it's spreading out throughout the area it's able to survive in. When it gets to this mountain range, one part of the population goes on one side and the other goes to the other side. And this is because they can't live at the top. So they're not going to be able to colonize the top of this mountain. And the populations are now separated so that the genes can't be swapped between salamanders living over here and over here. So this ancestral color has been passed down. When we get to this side of the mountain range, we're going to see more rocky environment. So we keep the same colors, but we see a more spotted pattern. So the salamander can blend in better probably with those rocks. As you go further south, the colors of the rocks shift and change. When you get to the south end of the mountain range, we see a different color of salamander. It's the same kind of pattern, but the colors have just shifted. And that actually doesn't take much of mutation to a gene for that to occur. And when you go even further south, we see just a different color variation and wider patches. These salamanders have been selected for based on being able to blend into this environment better than the ancestors could. If we go to the other side of the mountain range, we see this color pattern. It's the same as the ancestral colors, but a little bit more intense, a little bit more bright. When we go further south, you can see a very similar color pattern to this ancestral population, but we've lost more of the orange. So similar, but slight changes due to the soil conditions, the rock conditions on where these salamanders live. And their color patterns make them harder to see by predators. And so the ones that blend in better will pass on their traits. And this little population here has developed a lighter color than its ancestral population, still very similar, but very light to blend in with the sand. So these populations are no longer able to interbreed now that they have these color variations. They're just too different. Darwin spent a lot of time in the Galapagos, and he observed different populations of individuals on different islands. And he noticed that all of these finches were very different, but they looked similar to the finch found on the mainland. One of the main differences he noticed was their beak shape. And he noticed that the beak shape matched the food sources on different islands. Each island had a different food source, and the beak shape changed with it. And that helped him come up with his idea of natural selection, that things in the environment, these random changes, are going to select for organisms that possess variations of traits that help them to survive better. In the 1970s, two scientists, Peter and Rosemary Grant, went to the Galapagos to test Darwin's theory of natural selection with these birds. So in 1976, on one island, they caught 751 birds. They measured their beak depth, okay, so how thick the beak was, and they plotted it here on this graph. So we have the beak depth here, and we have the number of birds with that beak depth on the Y. So for example, here we had about 70 birds with a beak depth of about 9.6. Between 1976 and 1978, there's a terrible drought and a lot of plant species died, particularly those with smaller seeds. The ones with bigger seeds were able to survive. So in 1978, Peter and Rosemary Grant captured birds again, and all they could catch were 90 birds, and big drop in the survivor rate. And so when they measured the beak depth, they noticed that the average beak depth had increased from about 9.4 to about 10.1 millimeters. And that makes sense because the bigger seeds were the main food source that was left alive on the island. We also see less variation. So less birds with smaller beaks, less birds with bigger beaks. There are different types of natural selection where the environment will push for a particular variety of a trait. Stabilizing selection is what we just saw, where the selective pressure pushes toward a middle ground of the variation. The extremes of the trait are lost. Disruptive selection is when the middle is lost and the extremes are selected for. So if for some reason we lost the birds with medium-sized beaks and we selected for more birds with bigger and smaller beaks. So you get both extreme variation showing up and the middle variation is lost. Directional selections where we push for one side or the other, big beaks in birds or very small beaks in birds. There's also artificial selective breeding that humans will do. We select for the traits in organisms that we want to see stronger, and then we breed organisms together that have those traits. Sexual selection was also proposed by Darwin. Sexual selection occurs when these females in the population select for very flamboyant traits in males that we would think otherwise might be harmful for their survival. For example, these bright birds might be more visible to predators. We would imagine they would be picked off. But since they are able to survive predation and grow and have these really flamboyant traits, there's something about them that tells the female they have good genes to pass on. And so the females have selected for these more and more crazy traits that we see in males. Fossils can be dated based on where they are in the fossil record. So fossils found at the bottom of a layer of rock are going to be older than fossils found at the top. And this is just because of the way sediments lay down, oldest to youngest, one on top of the other. And we're able to use relative dating to compare fossils from all over the world. You might have new fossils in one place that you didn't have in a different place, but you might have some fossils that overlap in the two areas. So you're able to know which one is older or younger based on this fossil layer from somewhere else. And fossils show us that body plans are conserved over time. We see this segmented body being passed down. 4 legs has been passed down for a very long time now. Plants have very similar traits in their body plans that have been passed through generations. And we also see very similar reproductive mechanisms and with slight changes over time as well. Radioisotope dating is more precise than relative dating. You can actually get an age on a fossil based on the chemicals that it contains. So elements that are radioactive are going to decay at a constant rate. We call that their half-life. So there's a certain amount of time it's going to take for half of this uranium to decay. And after half of it has decayed, that would indicate that this rock is however old that half-life might be. As another half decays, you would add another half-life to that, another period of time. For example, carbon-14 has a half-life of about 5,000 years. So if we had 25% of our carbon-14 remaining, then we would know that fossil is about 10,000 years old. Different elements have different half-lives. Uranium has a very long half-life, so we would use it for fossils we expect it are very old. If speciation is occurring and populations are being separated and transitioning into new species, we should be able to find fossils that indicate these changes and these transitions between two very different groups of organisms. And we should be able to predict where we would find these fossils in the fossil record. So this has actually been done using transition fossils. This is Archaeopteryx. It's long been known that birds and dinosaurs have a lot of similar characteristics in their body plans, in their bone structure, in their skull structures, in the way they walk. So it's thought that birds were the species that developed from dinosaurs. And scientists predicted they would find this transitional fossil before they ever found it. And when they did, it was where they thought it would be. So this again is Archaeopteryx. It's got feathers. It's got the same kind of bone structure that our reptilian dinosaurs would have had. It's got the same skull shape. So this shows the transition between these dinosaurs and birds. Whale evolution is documented well by the fossil record. See what I did there? So we know that whales originated from a mammal with 4 legs that spent a lot of time around the water, probably developed syndactyly, where it was able to have webbing between its toes and swim better. Over time, longer limbs are going to develop with more webbing to help them swim. Those organisms survive and pass on these traits. So we have these very gradual changes to the skeleton over time, eventually losing these back forelegs. They actually are fused together in the skeleton of these newer whales, and we still see it in modern whales today. So the forelimbs shrunk into flippers. So this is mostly the arm bones are now inside of the whale, and these are very long finger bones that we would see here. But they still do have all of the bones seen in the forearm of a mammal. When we compare anatomical features like this, we can get clues to common ancestry and how organisms are related. We would expect 2 organisms with feathers to be more closely related than they would be to another organism without feathers, like a mammal. So birds more closely related to another bird than it is to your cat. When we compare the anatomy between 2 different species, we might look for these homologous structures. These are body parts that have the same structure but differ in their function. For example, the human arm, the cat forelimb, the whale flipper, and the bat wing. They're all completely different in their function and what they do for the organism, but they're put together in a similar way. They have the same numbers of bones connected in the same ways. The only difference is how long some of these bones are. The human arm has a longer humerus than the bat wing, for example. And the only way you would get these similar bone structures is if they had been passed down from a common ancestor and modified over time very slowly. So the more homologous structures 2 species share, the more closely related they are. And we see this body plan in all mammals here when it comes to the structure of the forelimb. Homologous structures would support the idea that 2 species are related. We can also look for analogous structures. These are parts of the body that have the same function, but are different in their structure. For example, the bird wing and the bat wing. There's differences in how these bones are put together. The bird has more fused bones than the bat does, and we assume that these bone structures are passed down from a common ancestor. Then it's been a while since these 2 have had a common ancestor, much longer between the bat and the bird than there is between the bat and other mammals due to how this bone structure has been passed down. So analogous structures do not indicate shared ancestry. So homologous structures indicate shared ancestry. Analogous structures did not. Vestigial structures are body parts that no longer play a role in modern organisms, but were very important in their ancestors. So the structure's been passed down, but its function has been lost over time. And these do indicate shared ancestry because that's the way we would have gotten this useless structure. For example, the human appendix and the rat also has an appendix. This is a little organ that hangs off of our large intestine. It really doesn't have a function in modern humans, so you can live without your appendix just fine. Many of you probably are living without your appendix. So functions are unclear in these vestigial structures. Another example would be the leg bones that we see in snakes. And these are now inside the body, greatly reduced, so snakes don't have legs. They've adapted to live without those, but they do still have a few bones from the legs that their ancestors had. And we can see this in larger snakes like boas. You might see a little bump on the side due to where these legs would have come out. Embryology is comparing embryos between different species. The more similarities there are in development, then the closer those organisms are related, because these development patterns have been passed down from parent to offspring over time. And if we look at embryos, no matter where the embryo came from, in these multicellular organisms, they all look really similar in early development. But as developmental time progresses, we see differences begin to emerge. The fish and the salamander retain a lot of characteristics that are similar, so they're closer related to each other than they are these other organisms. When we go to even later stage development, it's still difficult to tell the difference in these mammals. But now we can tell a difference in the reptiles and the birds, and we see that their developmental patterns are similar, so they're much more closely related to one another than they are these other mammals. And again, we're starting to see differences in the fish and the salamander, indicating splits in their lines as well. Biochemicals can also be used to indicate shared ancestry. Biochemicals are the molecules that make up life forms, the chemicals that code for life. So DNA, RNA, amino acid sequences, the closer these sequences are between two species, the closer related they must be. And keep in mind that DNA, that sequence has been passed down from cell to cell, parent to offspring throughout time as far as we can go back. So the fact that all organisms share DNA as their original protein code is a good indication that all life originated from a common ancestor billions of years ago. So biochemicals are good to analyze if you have two species that look closely similar, but they cannot interbreed. For example, we have a horse and a donkey. They look really similar. They can breed, but their mule that they create, that offspring, cannot have babies. And that means that they are too dissimilar genetically in order to breed. Without being applied, a theory is pretty useless. So we do apply this theory of evolution to health and medicine. If you want to test a new medicine on a certain protein in humans, then we would need to find a model organism like a mouse that would also make that protein and test our medicine on them first, and see how that medicine is going to interact with those proteins in order to know if it's safe for humans. So this is a table comparing the amino acid sequences of hemoglobin, the protein that carries oxygen in your blood. And these are the amino acid differences from human to human. There's really none. And these are the amino acid differences between these species and humans. So as you can see, gorillas have very few amino acid differences in hemoglobin from humans. That makes them a really good model organism to test new drugs on that might affect hemoglobin protein. Rhesus monkeys are pretty good. They only have 8 differences, and we see increasing differences as we go down. Another good one would be chimpanzees. They have very little differences as well. We also apply the theory of natural selection to agriculture when we think about how pesticides, herbicides, fungicides, all those things are applied on our crops, and fungicides even for some of our medical treatments. But these pesticides are going to alter the genetic makeup of the population. When we spray a pesticide or an herbicide, it's going to kill all of the members of that population that are most tolerant to it, which will probably be most of them. But remember, there's genetic variation in every population. Some of these individuals will be more able to withstand this pesticide, and they will be the ones who survive and reproduce, and they'll pass that trait on. So for example, here we have these beetles. We have red beetles and yellow beetles. These are just color coded. This isn't the color of the beetle. The red are more susceptible to our spray. The yellow are more tolerant to it. So when this pesticide is sprayed, it kills most of these intolerant individuals, and that leaves these individuals that can tolerate the spray to survive and pass that trait on. And if we keep applying this, we're selecting for more and more tolerance to this spray. So we have to change our pesticides and herbicides periodically. This is how we get things like super weeds that can't be killed when we spray Roundup on them. Aside from just chemicals we might come across in our environment, diseases are also apply selective pressures to a population. We're seeing this now with COVID-19. Diseases spread quickly in a population, especially when the numbers are high and individuals are densely packed together. So diseases caused by pathogens. These are disease causers, gen for beginnings of, path for disease. These are the beginnings of disease. Here we have a typical bacteria virus or a bacteriophage, and here we have the structure of a typical coronavirus, and they're called this because of these spikes on the outside. There are many different kinds of coronaviruses, not just the one that causes COVID-19. So these pathogens are spread around when populations are densely packed. And some individuals are going to be immune to this virus or this pathogen, and some individuals are going to be less tolerant to it. And we're seeing that with COVID-19, how some people show no symptoms at all, and other people are absolutely devastated by this disease. So immunity will build in populations over time as individuals who are less able to survive that disease die and do not pass that gene on, that susceptibility on. However, the stronger individuals will survive longer, pass on the trait to make the offspring more immune to that pathogen, and we'll see those traits accumulate. And there's been a lot of studies done on herd immunity in rabbits in Australia, for example, with this particular virus, but you can read more about those on your own. There's two kinds of immunity that we really see in nature. Active immunity is when the body produces antibodies, these proteins to mark a disease for elimination from the body. So these little antibodies are shaped like a Y. Mammals are really good at producing antibodies to new viruses. You may have heard some people produce antibodies to COVID-19 and some people do not. The people who do are getting active immunity. And you can have active immunity in one of two ways. You're either exposed to the virus naturally, or you get a vaccine that's going to expose you artificially to that virus and help your body get a kickstart in making these antibodies before there's enough virus to actually infect you. Once the body has made antibodies to fight against a particular pathogen, it's going to remember how to do that. So the next time you come in contact with that pathogen, the response will be a lot faster and you'll get better quicker. Vaccines can help with this because they'll stimulate the body to start making antibodies. The thing about vaccines is they will not cure you from a pathogen. They will not get you better from a disease or alleviate your symptoms, but they will keep you from getting sick in the first place. So that's why we get vaccines. And a good one to remember this with is rabies. We get our animals rabies vaccines so that if they come in contact with this virus, they're already making the antibodies against it. They're not going to get sick. Once a mammal has symptoms for rabies, there is no cure for that. It will die. There have only been 6 humans that have ever shown symptoms of rabies and been cured from that, but it required being put into a coma and long-term medical care. But now, if you are exposed, you think you may have been exposed to rabies, you can go get a rabies vaccine, and if you don't show symptoms for that virus, you're gonna be fine. So a vaccine will not get you better, but it will keep you from getting sick. That's a form of active immunity, your body making its own antibodies. However, another treatment of COVID-19 involves taking antibodies from those who have gotten better and injecting them into people who are still struggling with the virus. This is a form of passive immunity where you're taking antibodies from something else. Another really good example of passive immunity is breastfeeding. For about 6 months, while a baby is taking breast milk from the mother, she's passing antibodies to that child, so it's going to be immune to whatever she's immune to. After 6 months, these antibodies are no longer really being passed, and the baby's not getting any health benefits from that other than nutrition to grow. Breastfeeding, good form of passive immunity for very small children. We talked about how pesticide resistance and herbicide resistance can build in a population when we apply these pesticides and herbicides in agricultural practices. This can also occur in medicine with bacteria, and we can get antibiotic resistance. I've talked to you about plasmids, those little rings of DNA that can be passed to different bacteria. So bacteria reproduce very quickly. You can get E. coli reproducing every 20 minutes. So you go from 1 E. coli bacteria to over 1 million in about 6.5 hours. So bacteria really quick at reproduction. We would take antibiotics to kill those bacteria, but some variation is going to exist in this bacterial population through mutation. Some bacteria will be more resistant to this antibiotic than others. And this is why you have to take a 7 to 10 day treatment with your antibiotics. You want to kill off all levels of the bacteria. So these are again color coded with your resistance levels. Yellow has very low resistance. So after taking the first round of antibiotic, we kill off a lot of these weak bacteria and we leave the strong ones. They will survive and reproduce. So if you don't take all of your antibiotic, this can potentially happen in your body and you will retain those strong bacteria and they can continue to cause problems. And antibiotic resistance is a worldwide issue. These superbugs are becoming a big problem for treating. There was actually a lady who died in Texas a couple years ago where she picked up a bacterial infection from India that could not be treated with any of our known antibiotics, and she passed away. Antiviral resistance is similar. The virus is also going to mutate just a little as it's reproduced in the body. Remember that virus is not affected by antibiotics at all, but there are some drugs called antivirals that you can take. Tamiflu, for example, is an antiviral. But if these viruses are not destroyed by this antiviral, they can become resistant to it, and we can develop problems with how we treat these viruses. This also think about with Germ-X and how you sanitize your hands. It's better to wash your hands and just get the viruses and bacteria off of them than to expose them repeatedly to chemicals and build up this tolerance to your hand sanitizer. So if at all possible, it's better to wash your hands than to sanitize them. So that was a good basic coverage of evolution. Of course, if you have any questions, please contact me. Try your quiz on this. I do expect you'll see a lot of evolution on your final exam. And if you have any questions, I'm here for you. Bye, guys.